Hello grade 12 A-level students. Right now I'm going to discuss about the amino acid again from another textbook. Yesterday I discussed our textbook, and now today I'm going to discuss with the other textbook. Okay, this is still about amino acid, so your knowledge is getting enriched. Amino acids are amphoteric. They show both acidic and basic properties depending on the conditions. Means the pH of many body tissues is near pH 7. At this pH, both NH2 and the carboxylic acid of the amino acid are ionized. So at pH 7, an amino acid, example glycine, exists in a charged form. In the positive and negative charges are balanced. So like in this structure, in acidic condition, amino acids become positively charged. In alkaline condition, amino acids become negatively charged. Of course, in acidic, because it will lose a proton, so it will become positive. And then in alkaline, it becomes negative because it gains a proton. Okay, next, proteins are condensation polymers. On previous, based on our textbook, we learned that amine group of one amino can react with the carboxylic acid of another amino acid to form a dipeptide by condensation reaction. They will form an amide linkage. Eventually, a polypeptide containing many peptide bonds is formed. And then, when they are formed, polypeptide can contain 50 until 2000 amino acids. An amino acid unit within a polypeptide is called as amino acid residue. We draw the amino acid sequence in polypeptide starting from the end that has the free amine group, which is called as N-terminal, and then with ending with the C-terminal, which is the carboxylic acid. Proteins may contain one or more polypeptide chains. Some important facts about proteins are formed by condensation polymerization. The polypeptide chain in protein is unbranched, is different from carbohydrates. Each protein has a unique sequence of amino acids. As we know, there is thymine, guanine, cytosine, and then other things. Each protein has a particular biological function. Example, in the nucleus, it consists of proteins, amino acid that makes up the DNA. Now let's look at the primary, secondary, and tertiary structure of protein. Primary structure. Primary structure, the sequence of amino acid in the polypeptide chain. And then the secondary structure is a regular structural arrangement stabilized by the hydrogen bonding between the amide group of the peptide link. And then tertiary structure, the further folding of the polypeptide chain into three-dimensional shape. This 3D shape is stabilized by attractive forces and bonding between the amino acid side chains, the R groups. Each protein has its own unique function because each protein has its own unique 3D shape. This is the reason why it has its unique function, because it has its own unique 3D shape. What makes those unique shape? Because of the attractive forces and bonding between the amino acid side chains. Let's see the primary structure. The primary structure would be look like this. This is the example of the primary structure of the insulin A chain. a short polypeptide of 21 amino acids. So, okay, this one here is the N terminal, the amine. This one the carboxylic acid terminal. Okay, the primary structure, it is written as amino acid number N from the N terminal. Determines the way the protein can fold from its secondary and tertiary structure. It is held together by covalent bonds. So the bonding between them is covalent bond. There are S and S here. Sulfide and sulfide. Sulfide and sulfide is the active site. It is a disulfide bond. Why? Because they form a link. The link between sulfide and sulfide is stabilize the tertiary structure. Now secondary structure. Secondary structure describes the regions of the polypeptide chain in which there is a particular arrangement of the amino acid residues or single amino acid. The structure is stabilized by hydrogen bonding between the amide in amide linkage. The side chain of the amino acid are not involved. Two types of secondary structure. So in primary there is no side chain involving, but in tertiary there is a side chain involving. In secondary there is no side chain, it is only based on the amide link. There are two different structure, which is alpha helix and beta pleated sheet. Now let's see the alpha helix. Each polypeptide chain has a backbone of atoms. This is the backbone of atoms, CCN, CCN, that runs along the chain. Some points of the chains are flexible and allow free rotation around the bonds. The alpha helix backbone twists around a spiral so that a rod-like structure is formed. All the amide group of each peptide bond are involved in the hydrogen bond formation. The hydrogen bonds lie parallel along with the long axis of the helix with each NH group forming a weak intermolecular link to a CO group for amino acids residues further along the backbone. The large number of hydrogen bonds in the same direction stabilize the structure. So what stabilizes the structure? It is the hydrogen bonding. Okay, hydrogen bonding between them stabilize the structure. So this is the what it looks like. Okay, this is what it looks like. Okay, based on the structure. So based on the structure, this is what it looks like of the alpha helix structure. This is an alpha helix A. Along this one there is a backbone here, a stem, and then this is how the detail of the hydrogen bonding. The dot 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 means the hydrogen bonding. As you can see here, the red dots between O and H is the hydrogen bonding that stabilize the secondary structure of proteins. And then next, the beta pleated sheet. The beta pleated sheet hydrogen bonds are formed between the NH and CO group in different polypeptide chain or different areas of the same polypeptide chain. The figure 28.9 shows the beta pleated sheet in the structural protein silk. A fairly flat sheet like structure is formed. So the bonding between here is different from alpha helix, but they are what stabilize them is the hydrogen bonds. In alpha helix is vertical, but in beta pleated sheet is horizontal. It's between the oxygen and the hydrogen that form the hydrogen bond, which is the same as alpha helix, but the orientation of the bond is different, one vertical, one horizontal. Proteins may have a mixture of secondary structures. So like this one, which is very confusing. The structure, this model of an enzyme is pepsin. Pepsin is digestive enzyme found in the stomach. The structure, there is an alpha helix and there is a beta plated sheath. The alpha helix is the cylindrical rods that connects this stabilized by hydrogen bonding vertically, but for this one the arrow is horizontally make the hydrogen bond. Now we are on the tertiary structure. There are four types of uh, intermolecular forces that stabilize the structure. What are they? It is the disulfide bridges. Disulfide bridge. There's covalent S and S bond. Also in the synthetic rubber, and then there is also van der Waals, and then relative weak hydrogen bonds, and then ionic bonds. In this diagram, the textbook already the author of the textbook already give us information what is which one is the hydrogen bond, which one is disulfide, which one is van der Waals, and which one is ionic. So from this one, hydrogen bond is between the O and the H. Disulfide is between S and S in cysteine. And then in van der Waals is between the alkyl with alkyl. And then for ionic there is a presence of COO minus and NH3 plus. In this one, that make the intermolecular forces is the active site, not the amide link. Okay, so this is the difference. Disulfide bridge usually found in protein that function outside the body cell, example digestive enzyme. Disulfide bridge can form within the same polypeptide chain or between different polypeptide chain. The disulfide bridges help maintain the tertiary structure by locking the polypeptide chain in place. So what is the key is the disulfide in tertiary structure of protein. Dipole-induced dipole forces, or called as van der Waals, are formed when non-polar amino acid residues, the active site, are close to one another. A large proportion of amino acid residues in the center, many proteins are non-polar. So although the van der Waals forces are weak, the stabilization of tertiary structure due to total van der Waals forces may be considerable. And then there is hydrogen bonds formed between polar side chain having hydrogen bonds attached to a highly electronegative atom such as nitrogen and oxygen. So this one is the disulfide cysteine and cysteine. So they form disulfide bridge. Okay, and then this hydrogen bonding is between the hydrogen and the O. This is polypeptide. This is threonine amino acid. This one serine amino acid. So this one make a bond, and then this one is the ionic bond between two amino acids, between the two active site of the amino acids. And then this one make the COO minus and then also with the NH3 plus. Okay, those are the primary, secondary and tertiary. Now hydrolysis of proteins. As we learned on the previous part, hydrolysis of protein can be done by the presence of acid, aqueous alkali and also enzyme. The difference between them I already explained in the previous book. So we can do with reflux. So the condition is with reflux. With acid reflux, the product hydrolysis is original to amino carboxylic acid. The amine group will react with excess acid in the reaction to make a salt. With excess alkali such as sodium hydroxide, refluxing produces sodium salt of original carboxylic acid. So, for example, there will be a carboxylate salt, carboxylate ion. And then, so there is an importance of hydrogen bonding in DNA. We study already about the DNA. This is the pairing up. There is there are four types structure of DNA. DNA can make copies itself so that the genetic information can be passed on from generation to generation. And then DNA contains a sequence of bases that form a genetic code used to synthesize proteins. And then the nucleotides in DNA are made up of three components: sugar, called deoxyribose; phosphate, which is attached to the phosphoester linked to deoxyribose; and then nitrogen containing base. So this one is the main three component. There is a deoxyribose, and then there is a phosphate group, and then there is a nitrogen containing base. This one, guanine. Okay. So DNA, there are four bases: adenine, guanine, thymine, and cytosine. Okay. DNA consists of double helix. We know this double helix and alpha helix. And then DNA is kept stable by the presence of hydrogen bonding between the base pair. Okay, between the base pair and then van der Waals attractive force. What are the base pair? Adenine always pair up with thymine, and then guanine always with cytosine. That is this one, the base pair. Okay, and then you already know how DNA replicates when you study in biology, so I will not discuss more. What happens in the replication? The hydrogen bonds. So model of replication is shown. So this one is the steps. The hydrogen bonds and van der Waals between the base pair are broken. This part of double helix unwinds. The nucleotide triphosphate are brought up by one to the separated part of the chain, and then enzyme catalyze the polymerization reaction. So this one is the process. The hydrogen bond broken, the nucleotides, and then make a new bond. This one brought one by separate chain. Enzyme catalyze the polymerization reaction. So this is the process. Okay, and then this one, there is a polyester. Polyester is another type of condensation polymer. We ever discuss in when we discuss about the subchapter condensation polymerization. Condensation polymerization is always there is a characteristic of losing small molecule. Okay, so for today I already explained about the protein and then DNA and then also polyester.